people and as well as animals. So salmonellosis is caused by many serotype of Salmonella bacteria. Mainly the host is animals. So animal Salmonella reside in the intestinal tract of both warm-blooded and cold-blooded animals, poikilothermus. And in people, anyone who is infected with the Salmonella organism can get sick, but it mostly affects children who are small and immunocompromised people and elderly people. And Salmonella carriers often appear healthy. So transmission of the salmonellosis is mainly by eating contaminated food such as chicken, raw eggs, beef, milk products and vegetables. Any animal origin foods can be a potential source of infection to people. And pets, especially those with diarrhea, can pass stools containing Salmonella. So transmission to people occur when the people get contact with the feces of these animals, and if we doesn't wash hands and started cooking, and this can lead to transmission of disease. And person to person transmission can occur if infected people does not wash hands or if they do not follow the personal hygiene. So salmonellosis in humans, not everyone who is exposed to Salmonella will get ill. So it can happen to people. It will manifest itself as diarrhea, possibly bloody stools will come, abdominal cramps and fever between 12 and 72 hours after infection. And other clinical signs are headache, vomiting, diarrhea, dizziness, then muscle aches, etc. And without treatment, most people will normally become normal within a week, and it may take some people months before their bowel movements are completely back to normal. So in some cases, organism may pass into the bloodstream and be distributed throughout the body and causing organ damage, and this can become more fatal, leading to death. So this is the transmission of the and pathogenicity of the salmonellosis infection. So through fecal oral transmission, and there will be 7 to 14 days of incubation period, and this Salmonella bacteria will go adhesive to the mucosive invasion of epithelial cells. Then in the submucosa, it encounters to the dendritic cells, macrophages and enterocytes. From there, it will go attach to the infect the Peyer's patches in the small intestine and mesenteral lymph nodes. And from lymph nodes, it will reach to the reticuloendothelial system and finally to the bloodstream. In bloodstream, it causes secondary infections leading to bone marrow infection, liver, spleen and mainly gallbladder, which is a chronic reservoir of salmonellosis. And reinfection through bile excretion can occur in gallbladder. And severe complications include necrosis of Peyer's patches and toxic encephalopathy with myocarditis and hemodynamic shock. So heart will also get part of this infection, and which can become fatal. So in people, salmonellosis is diagnosed through laboratory test, culturing and serological culturing of the bacteria, and serological test can be done. And even in animals, the same method can be followed. Once salmonellosis has been identified, further testing to identify the serotype, so we can determine which antibiotic can be prescribed. So for the prevention, the person has to follow the personal hygiene, should wash hands with warm water and soap after handling with pest or their feces or using washroom. And all milk, meat products, everything has to handle them accordingly if it is contaminated, and should only buy pasteurized milk and milk products, and wash and properly store all the vegetables at proper time and temperature, and carrier animals must be identified and eliminated from a herd or flock. Last bacterial borne disease is shigellosis. So shigellosis is mainly a bacteria which belongs to the Enterobacteriaceae family. It is a genus of bacteria that is gram-negative, facultative and aerobic, and non-spore forming, non-motile, rod shaped, and genetically it is nested within Escherichia. So the genus is named after Kiyoshi Shiga, who discovered it in 1897. Kiyoshi Shiga is a Japanese physician and bacteriologist who first discovered shigellosis. So Shigella causes, thus the name Shiga came. So shigellosis causes diseases in primates but not in other mammals, and it is a positive agent of human Shiga, only naturally found in humans and gorilla, and during infection it typically causes dysentery. And it leads to bacterial diarrhea worldwide within 80,000 to 165 million annual cases and 74,000 to 60,000 deaths. So it is one of the top 4 pathogens that cause moderate to severe diarrhea, and it is divided into 4 serological groups which differ in the O antigen structure and virulence factors. That is type A Shigella dysenteriae, then S Sone, then S flexneri, etc. So it can be differentiated based on the biochemical metabolism, and S. flexneri is the most frequently isolated species worldwide, and S. dysenteriae is usually the causative agent of epidemics of dysentery, particularly in confined populations such as refugee camps. And Shigella chromosomes share most of their genes with those of E. coli K12 strain, which is MG1655, a well-studied model strain. And these Shigella strains are non-motile, and it is acid resistant, which survived our gastric pH 1 to 2 and which explains the low infectious dose. So virulence factors mainly include invasion plasmid antigen proteins and Shiga toxins. Shiga toxins is the toxin produced by the shigellosis, and it is a type 1 potent cytotoxin which leads to hemolytic uremic syndrome, which is a syndrome associated with low count of RBC, low count of platelets, and many kidney failure issues. And type 3 secretion system is a molecular mechanism followed which ingest this invasion proteins, IPA proteins, to the host cells so that pathogenicity can occur. And intracellular spread will be there, that is polymerization of host actin filaments allows the lateral spread from cell to cell. So foodborne transmission, outbreak strongly linked to person to food to person cycle, that is food handlers are critical in this infection. Common vehicles are salads, sandwiches and bakery items and milk products and contaminated water. Ice and beverages sold in unhygienic conditions are also a transmission vehicle. And Shigella does not multiply well in food but survive for a long enough to cause the infection due to its low infectious dose, and it is having a low incubation period. Pathogenesis. Shigella infection is typically by injection of fewer than 100 bacterial cells that may cause an infection. The species generally invade the epithelial lining of the colon, causing severe inflammation and death of the cell lining, the colon inflammation, producing the hallmark diarrhea. Even dysentery of Shigella causes infection. So toxins produced by some strains will contribute to the disease during infection, and both Shigella toxin and other toxins are associated with causing potentially fatal hemolytic uremic syndrome. So both E. coli and Shiga toxins are associated with causing potentially fatal hemolytic uremic syndrome. So it is a species which invade the host cells through the M cells. So this is the picture. So it is you can see that it is a toxin which invade the host through the M cells and intersperse in the epithelium of the small intestine, and it uses a type 3 secretion system, which is a molecular system that acts as a biological syringe to translocate toxic effective proteins to the target human cell proteins and can alter the metabolism of the target cell. and for example to the slice of the vascular membrane or reorganization of actin polymerization to facilitate intracellular motility of the Shigella bacteria. And inside the host cell, type 3 secretion system plays a crucial role. So this Shigella, after infection, the Shigella cells will multiply intracellularly and spread to the neighboring cells or neighboring epithelial cells, resulting in tissue destruction and characteristic pathology of sugar loss. And laboratorily we can diagnose it by culture based selective media like XLD, Hecton, McConkey was used, and biochemical profile, it is non-motile, urease negative and citrate negative, and serological assays is based on O antigen based typing, and molecular methods such as PCR for IPA, H and STX and virulence genes, whole genome sequencing increasingly used for outbreak tracing. And the main critical cause or main problem the world is facing, that it is having an antimicrobial the antimicrobial susceptibility testing is critical due to the rising resistance, the antimicrobial resistance developed by the strains of Shigella. So no licensed vaccines are available till now, but experimental vaccines such as live attenuated strains with weak strains such as flexinary mutants, conjugate vaccines which is targeting the O antigen, and subunit vaccines which IPA proteins and type 3 secretory system components are used. And development is complicated due to the serotype diversity and the need for the broad protection. So in order for the prevention and control of these diseases, prevention and control of foodborne diseases and requires a comprehensive farm to fork approach involving multiple strategies. So at the production level, good agricultural practice, that is GAPs, such as the use of clean irrigation water, proper waste disposal and control of animal reservoirs help reduce the contamination at the source. And in food industry, strict adherence to good manufacturing practices and exclusion of symptomatic food handlers along with training to prevent the cross contamination are necessary. And proper storage through maintenance of cold chain refrigeration, that is below 4 degrees, freezing and avoidance of the bacterial danger zone, that is between 5 to 60 degree, minimize the pathogen growth. Sanitation, proper sanitation and disinfection of food contact surface, utensils, equipments, and supported by pest control further reduce the risk. And mainly surveillance system including HACCP monitoring, microbiological testing and international network like PulseNet strengthen the early detection and prevention of the outbreaks. And consumer education, which plays a key role in the encouraging safe practices such as hand washing, thorough cooking and separation of raw and cooked food, and attention to labeling and expiry dates. In conclusion, we can say that foodborne bacterial infections and intoxications both remain a major global health concern or challenge, leading to significant morbidity, mortality and economic burden. So pathogens such as Salmonella, Shigella, Staphylococcus aureus, Clostridium botulinum and others can cause illnesses either through infection or toxin production, often linked to poor hygiene, unsafe handling, inadequate personal hygiene on processing. Effective control demands a farm to fork safety approach, so integrating good agricultural practices and manufacturing, that is GAP and GMP manufacturing practice, stringent surveillance and safe food handling and consumer awareness should be done. And with rising threats like antimicrobial resistance, there is a need for sustainable interventions, rapid outbreak response and international cooperation. So ultimately ensuring food safety protects not only individual healthy but also strengthen the global food security and public health system. Thank you.